Okay, so I think we have a few people making their way in, which is good. So maybe we can give it another sort of 10, 15 seconds. Luckily, we don't need to deal with any queues of people coming in. We can just let them digitally take their seats. Brilliant. We have people coming in. Fantastic. So hello, everyone. I hope you're, of course, very well. And welcome to today's webinar named Bridging the Gap, New Insights on Migraine from Pain and Headache Research. So, of course, June is Migraine Awareness Month, and we're supporting this with a webinar exploring recent advancements in migraine research. And we have some experts who you'll be able to see on your screen with us today, who will present and discuss two newly published studies from Pain Reports and the Journal of Headache and Pain. So I'll give our speakers a chance to introduce themselves just a little bit later. It's a little bit different to what we've got today. We've got some extra special guests. We're going to have an introduction from two individuals who from both EFIC and the European Headache Federation, EHF, about the organizations and to give us a little bit more of an overview. So first, though, before we do that, let's quickly mention how you can engage with the session. So like all of our webinars, there's a Q&A function at the bottom of your screen you should be able to see. Please use this to ask questions. It's obviously the benefits of a live webinar. You can also do this anonymously if you would prefer to do so, and we'll get through as many as possible towards the end. You can pop your questions in throughout the webinar, and we will get to them towards the end. There's also a chat function there that you can use for sort of general comments or general points that perhaps isn't quite a specific question. And obviously a reminder that this is a recorded webinar, so you can access these discussions and the content at a later time if you wish to do so via the EFIC Academy and, of course, many social media platforms that will spread this on. My bit's done just for the kind of beginning bit there. So I'd like to hand it over to our first guest, Antoinette. Welcome. I hope you're doing very, very well. And thank you so much for being here.
Thank you very
much. Would you like to briefly introduce yourself and give us an overview, an introduction to EHF?
Yeah, I will share my screen. And is it visible?
It is indeed. Thank you.
Okay. Yeah, my name is Antoinette Maas van den Brink. I'm the president of the EHF, and I will just shortly introduce the EHF to you. I'm a professor of neurovascular pharmacology at the Erasmus MC in Rotterdam. Ones of you who visited the European Headache Conference last year were in this city. The weather is now very nice in contrast to last December. And already over 30 years, I'm involved in research on migraine. I'm speaking here not from my affiliation in Rotterdam, but as EHF president. Here you can see the board. So Uwe Reuter is next president. And yeah, we have quite a tight team where we ensure continuity in our acts. EHF has published and is still publishing a couple of book series together with Spring and Nature. These are two books that I edited. And what I would like to highlight is the School of Advanced Studies, which is a nice initiative, a 2-day course on headache with each time a different program meant for junior headache doctors or basic science researchers. And admittance is free of cost. Coming October, we will be in Tallinn and look at the EHF website to see more information on the registration. Then I would like to highlight the future headache experts, which is a very important part of the EHF that we have since some years. It's a branch, especially for young headache doctors or researchers. And anyone may feel young, we define it as below 40 years. They organize webinars and events at our Congress. And in future, we also plan to make exchange programs, etc. You can apply via the EHF website. It's free of charge, or via the QR code that I show in this slide, but also the next slide. This is the current board composition, 5 young and enthusiastic researchers. And what does FHE do? I told you about seminars they organize. It's not top-down, but it's topic and speakers that the young investigators come up with themselves. And they organize activities at the Headache Congress. This was last year's headache conference and coming headache conference, which I will tell more about. They will be also active. So they organize graphical abstract competitions, scientific sessions, working group, meet the expert sessions, and FHE junior board members are also involved in the scientific program committee. Then I would like to draw your attention to the coming Headache Congress that will be in Lisbon. I really invite you to come there. It's from 3 to 6 December. And there we will, as usually, have a tough scientific program, social activities, and also a special get-together with our member states. And that's a nice bridge to the lecture that you will hear from Peter Pechner. Some weeks ago, I had the pleasure of visiting together with past President Christian Lampel, the Hungarian Headache Society, and it's really very inspiring to see all the activities that take place in the member states. So I won't take more time because the scientific program is what's most important in the lecture, and I hope to see you all in Lisbon in December.
Thank you so much, Antoinette. And it's fantastic to hear that I have at least a few more years of being young. We're not quite there. We are getting there very slowly.
And age is very relative. I still feel young, but I'm slightly above 40.
Slightly about 40. Brilliant. Well, thank you so much. Okay, let's hand it over to Sam. Welcome, Sam. It's great to see you. Do you want to take ahead and present on behalf of EFIC?
Thank you, Nathan. Sorry, first of all, apologies for my voice. I have a cold at the moment, so I may sound a little strange. And thank you to Antoinette for joining us and for collaborating. It's really important for the pain community to work with other organisations like the European Headache Federation and to build those bridges between different scientific societies, different disciplines, different fields of science. Pain is multidisciplinary. Pain is a shared responsibility across multiple professions, and it's really important that we have this collaboration. I'll just share a few slides on EFIC briefly. Tell me, Nathan, if this is working.
Yep, go ahead.
Yep. Okay, thanks, Nathan. So I think most of you on the call will be aware, but in case, in case not, my slides seem to have a life of their own. European Pain Federation is a federation of national pain societies, all of which are members of the International Association for the Study of Pain. Currently 40, with some recent chapters joining from places like Belarus and Georgia. We represent close to 20,000 healthcare professionals and researchers. So a lot of those are physicians, but also we are very multidisciplinary, including nurses, physios, psychologists, and others. And of course, those involved on the basic research perspective. And most importantly, we cover both the clinical management aspects, but also the research and cutting edge science aspects in this organization. We have activities in various fields, but I would say that the three main pillars of EFIC would be education, research, and advocacy. I've got the Congress here as a separate pillar just because it is the largest thing we do, like many other scientific societies. But it would be fair to refer to other activities that we do, such as our EFIC Academy project, which is our e-learning project. It's like an e-learning community that is thriving with around 1,000 members, with a lot of lectures and e-learning materials on the kind of foundations of pain science, but also more kind of advanced clinical management focused materials as well. We have exams for clinical professionals to demonstrate their skills and competencies and hopefully to have some kind of impact on their career development at national level as well. And those exams are becoming recognised in countries such as France and the Netherlands. In terms of research, the biggest project recently has been our pain research strategy, which we published recently and which we are disseminating right now. And hopefully that'll have an impact on the research community directly, but also on funders and policymakers. So that's something which we can direct you to if you're interested. We also do things like we establish best practices and develop evidence-based guidance for the clinical community. We provide funding for early career researchers. And in terms of advocacy, if you're not familiar, we have a very large advocacy project called the Societal Impact of Pain, where we try to share best practices, highlight the impact of pain, and on top of that, actually try and influence the policies that affect pain management and the lives of people living with pain. Um, just a couple of graphics just to show you here. So first of all, if you're coming at this from a headache perspective rather than a overarching pain perspective, you can see here on the left what we mean when we talk about chronic pain and the various pain types within that. So each of these is a specific field of study in itself, but we talk about chronic pain comprehensively. We of course deal with acute pain as well, but chronic pain conditions would be, the kind of the lion's share of what we deal with as a scientific society. And orofacial headache and orofacial pain being an important category within that. And we also talk about pain as a biopsychosocial concept. So you can see here on the right a kind of visual explainer for the different factors that influence the pain experience. And then finally, I'll just say that our next Congress is in Glasgow in 2027. So we're very early in the cycle for promoting that. But if you're interested in attending a pain science congress and you haven't before, please bookmark these dates. Okay, on with the science, and I'll pass back to you, Nathan.
Yeah, thanks, Sam. Really useful information. And again, if you need to access any of that, the EFIC website or reach out to anyone at EFIC will help you. If you have any questions for Antoinette or Sam, please get them in as soon as possible. These guys are incredibly busy in trying to access their big, insightful brains. It's actually quite difficult. So if you have any questions, please pop them in as soon as possible because they do have other meetings and things to get to. They don't get to stay for the exciting stuff. But yes, thanks, guys. Really, really valuable. So now let's move on to our speakers for today. First, we have Peter. We're very interested in what Peter and a little bit later Campo have to offer for us. So let's hand it over. Good morning, afternoon, Peter, and thank you for joining us. Please go ahead with your presentation.
Thank you very much, Nathan. Just let me share my screen. Okay. And now you should be able to see the first slide, right?
We can, thank you.
Okay, hello everyone, and really thank you for having me. It was a great honor to be invited and speak about our recent paper entitled The Interictal Transcriptomic Map of Migraine Without Aura. Actually, I thought that most of the participants will already know about migraine, what it is, so I skipped that part. But I assume that maybe what I have to talk a little bit about what are RNA investigations and transcriptomic investigations. So as you can see here, most of the time from a research perspective, if we want to discover some kind of pathomechanism or drug targets even, then we are interested in proteins. But generally proteins are hard to measure in a systematic way. So for decades now, there is a methodology to do RNA-based investigations. And kind of we assume that RNA levels correspond to protein levels eventually. Of course, that's not 100% match, but they correlate well enough to have some inference about the background of a phenomenon biologically. So since RNAs are easy to measure, I mean, easy is of course relative, methods were here for decades to measure RNAs in a systematic way. Previously, microarrays were a kind of mainstream method using hybridization. And today, essentially sequencing of RNAs, especially messenger RNAs that have the quality to inference about protein levels, are measured by mRNA sequencing. And of course, in migraine, there were also previous attempts to recover some kind of transcriptomic map or RNA map of the disease. So there were quite a few studies that tried to investigate them. But when we looked at the genes, essentially these studies could not really replicate each other on one hand, and also on the other, in many cases, they could not really discover really significant genes. What I mean by really significant is that if we measure a lot of RNA agents like mRNAs, then of course we perform a lot of hypothesis tests. So we need to correct for this kind of statistical bias. And we usually use some kind of statistical correction for that. So what I mean that these two studies, for example, could not recover such strong candidates, gene level candidates in migraine. There were other studies as well for chronic migraine, for example, or mRNA expression during attack versus the interictal period. But that was not our main purpose, so I do not really discuss them now. And also for RNA sequencing and also for microarrays in general, you can recover pathways as well with the method by simply aggregating the measured mRNA levels in a sense onto a pathway level and calculate the statistic on that one. And also these studies try to do that, but essentially there were very non-informative pathways for migraine. Immune pathways were something, or inflammatory pathways were something that kind of popped up regularly in these studies. But other than that, there were no trace of known migraine pathomechanisms and that were previously proposed by other methodologies. And we wanted to do that. I mean, we wanted to recover some kind of beta mechanism. We even wanted to recover some kind of novel drug targets if possible. So we wanted to do it with our study. And we had some key ideas what may have been the cause behind kind of such scarcity of results. And so we tried to correct these aspects and after collecting them, essentially. The first thing we wanted to do is to collect a large clinical sample and restrict actually migraine patients to migraine without aura. So the one of the subtypes of the disorder. And we could collect actually 30 control patients and 22 migraine without aura patients who were diagnosed by neurologists. And the other thing that in order to have actually this kind of statistical power, and the other thing we wanted to do is, or we assumed that may be the case behind the heterogeneous results and thereby the lack of statistical significance in genes was that we assumed that actually people or migraineurs with comorbidities and medication intake might be actually or might show different mRNA patterns than without these comorbidities or medications. So we wanted to really collect a very homogeneous sample of patients. And we tried to do that, actually. We collected much more people, that only these ones remained for the RNA sequencing. And there were only two factors remaining. So we excluded everybody with any other comorbidity except migraine and except allergy, because it was so comorbid that if we would have excluded allergy, we would have excluded almost all our patients. And also contraceptives, because that was something that many of the patients were taking. So these were two factors, but we also recorded these covariates and we wanted to essentially correct for them during the analysis step. So in addition to correction for age and sex, we also corrected for smoking and allergy status, because we assumed that they may also influence the measurements. And we also wanted to replicate actually measurements within our own study. So we called back the people and essentially we replicated all the measurements with an average time difference of 4 weeks to really report only stable findings that we thought that they might be reliable enough to use in future studies. And eventually, of course, we assume that we find something, so or we will find something, and then we wanted to do some functional analysis, like genomic background of the visible mRNA changes. There are polymorphisms within them that may explain for this, or drug binding studies, etc. So we use machine learning and also GWAS study we conducted GWASs to essentially kind of identify the background of the observed mRNA changes. So this is the whole scope of the paper. That's one of the figure of the paper. And I will only talk about basically the core results and some of the pathomechanism and causal factor identification. We tested a lot of things. As I mentioned, we also wanted to do drug candidate identification or novel drug candidate identification. So we did it. I don't talk about that here because their time is very limited. Of course, you can imagine the surprise that what we saw when it turned out that we still were unable to find replicated significant genes actually on a gene level. I have to mention though that actually at one time point we had approximately 5, if I remember correctly, 5 significant genes, but essentially since our criteria were that we wanted to report only replicated significant genes. Actually, they didn't replicate in the second measurement, so we could not significantly. So we could not say that these are replicated and significant genes. And also, I have to mention that without allergy correction, essentially, we had a significant gene. So it showed how influential allergy comorbidity can be, and probably that there's really a tight comorbidity and shared pathomechanisms behind disorders. Nonetheless, we have found a lot of pathways that were significant and replicated, and they were really related to known migraine pathomechanisms. So, for example, as I mentioned already, immune system-related pathways were already raised as kind of candidates, and we really found a lot of them. But also others were popping up, like cardiovascular, stress-related pathways, metabolism, carbohydrate metabolism, amino acid metabolism, and lipid metabolism, all of them, and even hormone response as kind of pathways. And we collected here, so these yellow kind of, I don't know, baskets, they are summary terms for these pathways, but there were a lot of different pathways in each category I mentioned here. And then we went further, and there is a type of genes behind these pathway level analysis. These are called leading edge genes that were driving the pathway level results. So you can imagine it as like these genes were the main factors why a pathway became significant in our studies. And there were 2 really interesting candidates in this behind these pathways that we have found. One of them was chorin, that is a serine peptidase, and the other one was CYP26B1 that pointed to retinol signaling. And essentially, we went further to analyze the background of these genes, why they might have popped up. And we did also retinal pathway analysis, and we saw that nominally significantly downregulated retinal pathways were coming actually. So there was an underlying not completely significant, but still some kind of indicative result in the pathway level. And we also analyzed these LE genes, so these leading edge genes, whether they are regulated by retinol receptors, RXR and RAR. And actually, we found that many of the leading edge genes were regulated essentially by retinoic acid receptors. And we also did a GWAS analysis, and we found that on one hand, migraine patients took less vitamin A in general, but on the other, actually, LRP1 polymorphisms that have been implicated previously by other genome-wide association studies, and this is also involved in retinoic acid uptake. Well, these polymorphisms significantly associated with migraine in both the UK Biobank, really large cohort, and in our own cohort, and that was independent of the different vitamin A intake. So this kind of suggested a common pathomechanisms that we could see. And that's the final slide, I think, about that. So what we assume, or what our kind of what our article proposed, is that there is a different LRP1 genotype in migraineurs that leads to currently unknown mRNA and protein level changes within the LRP1 that we were not able to detect because LRP1 was not expressed in blood, so we cannot see that. And we measured our mRNAs in blood. And that leads to an impaired chylomicron remnant uptake by LRP1, and thereby actually an impaired retinol transport to the periphery, which then causes a lot of different pathway level changes. They could actually almost all of our pathway level changes could be bound to some extent to retinol signaling. And also, actually, there was a maybe Nature publication showing that this LRP1 is also involved in, from a different perspective, into myeloid cell retinal uptake. So it also can influence the immunologic mechanisms that were recently discovered, or not only recently, but previously discovered in migraine. So that is kind of one message of our paper, but of course there are many, as I mentioned. So there are other drug binding simulations as well, etc. But I wanted to share with you this one. And of course, we have a lot of other studies ongoing. So this is Gabriela Juhas in the center, because she coordinates all the migraine-related studies, and we have other publications as well related to migraine. You can see here some of them. These are my PhD students, Dora and Zahel, with whom I also worked on the current paper published. And these are my acknowledgements I have to share. So essentially, and thank you very much for your attention. I hope I could say something new to you as well. Thanks.
Absolutely. Thank you. Thank you so much, Peter. Appreciate that very, very much. Would you be able to just briefly give us a bit of an introduction to yourself, your institution, because there's a lot of information about the work that's going on. Tell us a bit more about the institution and a bit about your background.
Okay. So I work actually right now. I'm an assistant professor at Semmelweis University in Budapest. And during this work, I was also working in Japan at the Bioinformatics Center at the Kyoto University for 3 years. So I'm kind of, in that sense, bioinformatician. And I started my PhD in 2010, I think, with transcriptomic investigations in depression. And then I actually kind of got involved in genomic investigations as well, in depression and in migraine as well, because they are very highly comorbid. So it was a kind of trivial thing. And there were a lot of joint research going on here. And yeah, basically, that's it, I think.
Just that's it. That's it. No, thank you so much, Peter. Appreciate
Thank you very much.
Okay, let's move on to our second speaker. Kwampo, welcome. Thank you so much for spending time with us today. Looking forward to the presentation. I'll hand it over to you to share with us today your thoughts and your experiences, and of course, an introduction to yourself and your background will be really useful. Thank you for joining us.
Thank you, Nathan. Thank you for having me. My name is Kwampo Pan. I'm a neurologist based in Hamburg, Germany. And I'm now trying to share my screen. I hope you can see it well.
We can indeed. Thank you.
Yeah. So today I'm going to share with you one of our recently published paper with the title Cycling Sensitivity Across the Migraine Phases, a longitudinal case control study. And this study was recently published in the European Journal of Pain. And this was actually a proof of concept because we had a hypothesis back in like I think 4 or 5 years ago, and we tried to prove this hypothesis, and here is the results. And first, we talk a little bit about the background. What is a migraine attack? It might be a simple question. Many of us, we just think that the headache is the migraine attack. It's something we have been aware of for decades or even for thousands of years. But accumulating evidence suggesting that not only the headache, because many migraine patients, they have the other symptoms like the prodrome symptom, mood changes, neck pain, sensitivity to light, to sound, food craving, nausea, yawning, and all these symptoms happen before the headache starts. So that's during the pre-ictal phase or the premonitory phase. So if we put all the different symptoms into the migraine cycle, traditionally we'd only talk about the migraine headache. That's the ictal phase. But with accumulating evidence suggests that the migraine attack is actually covering the pre-ictal phase, the ictal phase, and also the post-ictal phase, all of them. And there's also some studies suggesting that some of the symptoms the patient experienced in the pre-ictal phase, like photosensitivity and phonosensitivity, they are highly associated with the symptoms experienced during the migraine attack. That means that for those who with the photosensitivity in the pre-ictal phase, they are more likely to have photosensitivity, photophobia during the ictal phase. That suggests all the symptoms, they are like connected to each other. It's a continuing spectrum of different symptoms. So not only the ictal phase is important for the migraine attack, we have to cover the pre-ictal and also the post-ictal phase. Based on our knowledge about the symptoms, we came up with this idea, this hypothesis back in 2019. We believed all the symptoms, the cycling symptom is based on the cyclid sensitivity to different stimuli. That's the threshold theory. Because the migraine patient, they may have the altered threshold starting in the pre-ictal phase. That's the reason they are going to experience something as a little bit too much. For example, that means that the patient's sensitivity is actually decreasing as a result. A normal light would be perceived as too much during the pre-ictal phase and during the ictal phase. So if you want to know where did the migraine start, we have to focus on the start of the pre-ictal phase. Here is where it starts. And of course, for the healthy control, they also have the threshold to sensitivity, to light, to sound, to pain, but there might be some day-to-day changes. That was our hypothesis back in 2019. There were actually some studies supporting this hypothesis back then. There was also our review 4 or 5 years ago. But during that time, not many studies focused on the pre-ictal phase. Most of the studies either focused on the migraine versus healthy control, or migraine only in the ictal phase versus the interictal phase. And among the 5 studies here, you see that 2 out of 4, they actually look into the pre-ictal phase. And 2 out of 4, they can show that the pre-ictal phase, the sensitivity is already changing. But it was still like a mixed result, 2 positive, 2 negative. And we believe that one of the main reasons for the discrepancy among the studies back then, it was the study design. Because most of the study back then, they used a cross-sectional design. And I'm going to tell you why cross-sectional design might be problematic. Here is our pilot data. We look into 8 healthy participants, and we measure the pain threshold every day. And you can see here, each different color represents one single subject. And you can see the interpersonal differences is huge. Some of the participants, they have the electrical pain threshold up to 4 milliampere, and the other one is like 0.4. But if you look at how the curve changes over the 5 consecutive days, I would say that the intrapersonal differences, the longitudinal difference, is relatively not so much in comparison to the interpersonal differences. So in the cross-sectional design, as I just showed you here, it could be for the interictal phase, you capture some participant with a higher pen threshold, and for the interictal phase, you're capturing the other ones with a lower threshold. So it makes the comparison between the different migraine phases really difficult. So ideally, to look into the premonitory phase or the preictal phase is also really practically difficult. That's the reason why people do the cross-sectional design, because you never know when the patient is going to have the migraine attack. Of course, you can put them in the scanner and do something 30 days in a row, but it's not that efficient. So how are we going to predict migraine attack? What should we do to be able to capture the preictal phase to see what things is going on during the preictal phase? So the ideal candidate for our study will need a cohort with a low frequency migraine. And I'm going to explain to you why low frequency is really, really important. And also we need something as a predictable migraine. And the critical part is with that we can capture the preictal phase. So why low frequency migraine is critical? Because here is our simulated model. You see 4 different colors. And the blue one is the data is like simulated for 1 month. The blue one is for one single patient with only 1 attack per month. And you can see, I'm trying to use the pointer. For this attack, you can see the threshold go down and afterward it goes back up. And for a single attack, the preictal phase, the ictal phase, and the postictal phase combined together, it is at least 5 days. And if you put the red line here, that's the patient with 4 attacks per month. You can see after the attack, they still go back to the baseline. But starting with like 8 attacks per month, the green line, you see the baseline is like reduced. You don't come back to the baseline. And if the patient got 15 attacks per month, they are almost like continuously in the ictal phase. The pen threshold is not really changing. So that's the baseline for 1 or 4 attacks per month. That's the baseline for 8 attacks per month. And that's the baseline for 15 attacks per month. So low frequency is really important. If you include a patient with chronic migraine or patient with high frequency episodic migraine, it's really difficult to see the interictal and ictal changes because these patients, they are almost continuously in the either preictal, postictal, or the ictal phase. So ideally, to fulfill all these criteria, that's the reason we look into the menstrual migraine, focusing on both pure menstrual migraine or menstrually related migraine, but they must have only 1 extra menstrual attack at most. And here is our study design. Because for this cohort, we collect 3 months of the menstrual diary. We know almost when they are going to have their next attack. And based on their expected next attack, we plan our study design. It's like starting 7 days before the menstruation. So theoretically, we are going to capture the migraine attack somewhere here. And every day, we just did the same thing. We measure different kinds of threshold, including electrical, heat pen, cold pen, and mechanical threshold, every day until they have the migraine attack. Of course, one would argue that we are looking into the menstrual migraine. So if we found any changes here, it could be secondary to migraine or secondary to hormone changes, because we know that before the menstruation, there are lots of the differences in the hormone. And that's the reason we also have to include the match control cohort also every day until they have the menstruation. But this cohort, they are the healthy cohort, they don't have the headache. If we can find anything only in the migraine cohort, but not in the control cohort, we can say that the changes might be secondary to migraine, not secondary to hormone changes. And here is our results. And both of the cohort, they are matched in age, sex, and the menstrual cycle is also similar. And for the migraine cohort, they have average 4 days of migraine attacks. And most of the migraine days is only during the menstrual period. And actually only 2 out of 10, they have menstrual related migraine with 1 extra menstrual attack. And 8 out of them, they have pure menstrual migraine. And in our retrospective questionnaire, all 10 of them, they reported premonitory symptoms with an average number of premonitory symptoms up to 6. So first, the behavior data. For each of them, we measure 7 to 11 days continuously. And all the 10 patients developed a migraine attack. And all the attacks, they were migraine without aura. And to the baseline, 4 out of 10, they have migraine with and without aura, and 6 out of them, they have migraine without aura. But during the 7 to 11 days of consecutive measurement, all the attacks during the periods, they were attacks without aura. And every day we ask them, do you have any symptoms, something wrong? Do you feel anything different? 48 hours before the attack, none of them reported any prodrome or typical prodrome symptoms. And 1 day before the attack, 3 out of 10 reported some typical prodrome symptoms. And now we come back to the major results, the electrical pain threshold. On the right-hand side, you can see our model 5 years ago. And here is the results. The data is already normalized to each individual to the ictal phase. And you see that in the ictal phase, the pain threshold is relatively high. And in the pre-ictal phase, the threshold is already decreasing. And it is the lowest during the ictal phase and gradually recovered in the post-ictal phase. Here, the standard deviation is higher in the post-ictal phase because our design was also, we only plan to measure it until the migraine attack. So we accidentally captured 4 out of 10 post-ictal phase because they had the migraine attack on day 5 or day 6, and day 7 was already the post-ictal phase. So that's the result in the migraine cohort. And now we move to healthy control cohort. And we also have the hypothesis they might have the day-to-day variation, but not like something we see in the migraine cohort. And here is the results. Up to their menstruation, there's still some day-to-day variation, but the variation is comparatively less compared to the migraine cohort. The migraine cohort is almost like 200% variation. And for the healthy control, they have day-to-day variance, but it's relatively stable. And the next question is that we know that the migraine cohort, they have the not normal pain threshold. So is this the interictal phase of the migraine cohort not normal, or the ictal phase of the migraine cohort is not normal? So we use the raw data, not normalize the data, and compare both of the cohort directly. You can see the interictal phase of the migraine cohort is comparable to the healthy control cohort. So actually, it's the pain threshold gradually decreasing towards a migraine attack. And the baseline between the two cohort is quite similar. And actually, we also, I just mentioned, we measure not only the electrical pain threshold. Here we have the heat pain threshold, we have the cold pain threshold, mechanical pain threshold. For the heat pain, for the cold pain, we also found something similar. For the mechanical pain, it's not significant, but there's also a similar trend. The threshold is going down all the way toward the ictal phase. But the standard deviation for the mechanical pain threshold is too high. I would assume that we need a much larger sample size to show anything with the mechanical pain threshold. So to summarize our study here, first, as a proof of concept, we now shown with clinical behavior data that the migraine is driven by cycling sensitivity changes, as demonstrated by the threshold changes. And also the migraine attacks, also the sensitivity change actually started 2 days before the headache. And something really interesting here, because if you look at the data again of the retrospective questionnaire, all of the participants, they reported premonitory symptom, at least in their retrospective recall. but prospectively, only 3 out of 10 can actually report the premonitory symptom that leads to the migraine attack. 7 out of 10, they notice no symptoms. So I think here we have to make a distinction between the pre-ictal phase and prodromal phase, because both of the terms that are used to like mixed together oftentimes. Pre-ictal phase is simply for the time period that is before the migraine attack. And prodrome, it means that the patient has to experience certain symptoms, and these symptoms sort of predict the incoming migraine attack. We would argue that pre-ictal phase would be the more appropriate term for this phase, because most of the symptoms, most of the subjects, the patient, they didn't notice any symptoms, but they already had a decreased pain threshold. That means something was already going on, but they didn't notice that. And the prodrome is probably just represented because we show that the threshold go all the way down to the migraine attack. When the sensitivity is going all the way up, when the patient is really, really close to the migraine attack, to the ictal phase, to the headache phase, they are starting to notice something, some neck pain and some other symptoms. We believe that's the prodrome simply represent the gradually accumulated sensitivity. And of course, this study got some limitations. First, we got this from our reviewer. There's a small size study. Even this small size, only 10 for the patient cohort and 13 for the healthy control cohort, it was already 180 days of consecutive measurement. And we propose that the threshold model may apply to different kind of sensitivity, including photosensitivity and phonosensitivity. But in this study, we only test the pain threshold. It would be nice to have the other non-pain threshold included in this study. And also the minor differences between the non-menstrual cohort and the non-menstrual migraine attack and menstrual migraine attack cannot be excluded because we only studied the menstrual attack in this study. So based on the results of this study, there are some future perspectives. On the right-hand side is another study from our group. In this study, the researchers focus on the visual after image duration. And my colleagues can actually show that the after image duration was already changing 2 days before the migraine attack. And that has something to do with the visual cortex sensitivity. And here is another functional MRI study. Another colleagues of mine has shown that the hypothalamus has been activated also 2 days before the migraine headaches. So if the pain threshold changes as we identified in this study started 2 days before the headache, that is corresponding to both the functional MRI study and also the physiological study. Our next step would be to combine the threshold measurement and also the functional MRI study to visualize where in the brain is determining the threshold changes. And that's something we have been working on for a few years, and hopefully I can show you some data by the end of this year somewhere else. Yeah. So thank you for your attention. Let's move on to the discussion.
Yes, thank you, Kwan. Very, very interesting. It's great to see that you've been working, obviously, since this body of work to bring out all this new knowledge, especially regarding the fMRI stuff, which I think will be super interesting. So thank you for sharing that. Just a quick reminder, obviously, to share your questions and any thoughts that you might have within the Q&A and the chat function. You can, of course, do this anonymously as well, which I highly encourage, given that trying to send an email to these individuals can be quite difficult. Everybody's busy, so this is a great opportunity to ask a question here and now. Let's go into a couple of questions. We do have a good amount of time, actually, about 10 minutes. So we have one question, which I think, Kwan, you did allude to, and please feel free to add if you feel like you haven't sufficiently answered this question. I think it's directed perhaps more towards you, is how do you do quantitative sensory testing for migraine patients? How can one go about that?
It's actually a well-established method to test the sensitivity. There's 11 parameters, and like including vibration, heat, cold, and also mechanical sensitivities. And I did not mention where did we conduct the pain threshold measurement. We actually measure here on the forehead, and because our hypothesis was that the migraine is definitely a pain in the head area, the trigeminal system, not a pain on the arm. So we did not measure the arm. All the data I presented is here.
Okay, that's really interesting, and hopefully that's answered the question. Feel free to, there we go, fantastic, way ahead of me on sharing the screen. Peter, are you still here? If you are,
sure.
I think we have, there we go. A little question for you. You mentioned one, and you did mention a couple of times, there's lots of findings within the paper that you just didn't have time to share today. You mentioned the pathways, which I think is something that's really important for, or the potential pathways for our audience. In your opinion, what are the most kind of promising pathways that you feel warrant more attention in terms of guiding future research, or even guiding the attentions of clinicians? What were you interested to see in those pathways, and what do you think would be most interesting to kind of explore, and why?
That's a really good question. Just let me think a little bit.
No, of course, yeah. I think it's important to think about how the direction of the research is going, and it was a really good way, and I really like that graphic. It's nice and busy, but it shows very clear kind of directions into the different considerations, right?
Yeah, actually, what was interesting for me, and I think that is really worth to explore, was this kind of CYP26B1 gene, which is, kind of unified many of the pathways. I mean, it's related to this vitamin A metabolism. And as I mentioned, basically in the background, all these are really a large amount of genes, approximately 60 to 70% of the genes that we found behind these pathways were regulated by vitamin A related receptors. And, just a recent study came out in, I don't remember which journal, but they actually, they kind of looked at on carotenoid intake in migraineurs, and they could prove that essentially those who were low on that were more prone to migraine attacks. And what is interesting, that carotenoids are actually proforms of vitamin A. So essentially, I think previously, the same thing happened to vitamin D, that people were thinking about only a vitamin in itself. And then it turned out that it is a pathway in general. So there are a lot of things influenced by this one factor, including immunologic and all cardiovascular, etc. So, and that is exactly what we could see in the study, that this is kind of connecting these dots. And I think maybe that would be nice to see more studies, more mechanistic studies on vitamin A or carotenoids that are kind of prospective, so not like ours, that to test this idea of influence of the influence of carotenoids and vitamin A and retinol in general.
And it's always nice to see things replicated in other pieces of work, and you can see the directions and the links there. Kind of know you're along the right lines there, and of course this synonymous approach to research, and everybody kind of finding these little pockets of related findings to build our understanding is always really good to see. Fantastic stuff. Just a last reminder, bearing in mind we have 5 minutes left of questions into the Q&A. I won't remind you again, because you're probably annoyed at me reminding everybody, but you do forget when you're deep into the conversations. Quan, are you still here? Fantastic. We're all too courteous turning the cameras off. Feel free to keep your cameras on just for the remainder. Thank you for sharing that. And of course, I have put the papers for both of these presentations into the chat, so feel free to explore those as well. I kind of got the understanding, and I've learned a lot today, so thank you to both of you for your presentations, that although it's quite difficult to identify, it seems that the ictal and pre-ictal are getting better defined, and you're certainly advocating for this the distinction between the two. But I noticed before that the interictal phase, and I'm just trying to think from clinical practice perspective, what's kind of the most difference in terms of a clinical presentation between sort of the preictal and the interictal, the kind of two phases before that, and is that really difficult to see in clinical practice? You mentioned that some of the symptoms might not come in until later on in the situation. They come on very, very quickly. I think I saw you had 48 hours where there was none-report, and then 24 hours, all of a sudden you have them coming through. Thinking of the healthcare system, one day to try and support this management. And of course, I know a lot of migraine's support in terms of the self-management, but how difficult is that to identify? And is it about helping people identify those phases and when they start? Is that kind of where that's very interesting?
I would say for the individual level, it's really, really, really difficult to identify it properly. As we have 10 participants, all of them reported prodromes for their preictal phase. But actually, during the consecutive daily measurement, only 3 out of them, they actually have the symptoms one day before. So it's like, if you look at retrospectively, okay, I probably have this, that before my attack. But if you really use prospective the method to look into it, it's not necessarily reliable. And sometimes you get a false alarm. You didn't sleep well last night and you get tired, you get lots of yawning, anything. It might be a pre-ictal phase and or the prodrome, but it was actually nothing to do with the migraine attack. So I think what's really important is not to predict it. What's important, the message from our study is like something was already going on even up to 2 days before the migraine attack. That's contrary to the main vascular hypothesis for migraine, because the vascular hypothesis focuses exclusively or mostly in the headache. All the peptides, they have something to do with the headaches. But if something was already going on like up to 2 days before the attack, there must be something in the brain is building up for the migraine phase. I think that's the main message from our study. Yeah.
Fantastic. And it's widening the approach and the understanding of what's going on, and moving away from symptomatology and into these other kind of areas, which is very interesting to me. So thank you. Okay, I think we have time for one more question from the chat, which is brilliant. It says, is that or would that be the same region for trigeminal neuralgia as well on the forehead? Is that the same sort of position that you would measure? Is that correct?
We measured actually the V1. Is not ideal for headache because headache is the V1 in the dura, not V1 here. But for trigeminal neuralgia, it's more commonly V2 or V3 or both of them. I would argue that if somebody is trying to do a study with trigeminal neuralgia, I would definitely not pick the V1. Because we also have data that V2, V3, they are somehow different compared to V1.
Okay. Fantastic. There we go. There's the answer for you. Guys, thank you so much. I think this is a good time to bring it to a close. Peter, Kwampo, thank you so much for spending time with us today and sharing your work. Of course, everybody go and see the research that is in the chat. Of course, as well, thank you to Antoinette and Sam who joined us earlier, and thank you all for your time today and sharing some very interesting work. As mentioned before, this webinar is recorded, so you can access this via the Ethic Academy platform, YouTube and social media. Perhaps EHF might be able to share this as well, so you might see it on their kind of outreach as well. So don't forget to share this online so that others can benefit from this, of course, really interesting discussion. Thank you again for the questions and the introduction from the audience. It's been very, very fruitful. I guess it's all for me to say is I hope you have a fantastic day, and I hope to see you all on the next webinar. Thank you very much, guys. Appreciate it. Take care.
Thank you.
Thank you very much.
So,
Bye-bye.